right, so good morning, everyone. Welcome to another broadcast with us here at Exploring by the Seat of Your Pants. If you're joining us for the first time today, we bring conservation, adventure, and science into classrooms around the world through over 40 live monthly broadcasts with some of the coolest scientists and explorers on planet Earth. Today, I'm really excited because we are diving back in with our Gardner Foundation series. So this is our penultimate program, our sixth of 7 amazing featurettes featuring some of the coolest scientists in biomedicine, health, and technology in the world. Every year, we get the privilege of partnering with the Gardner Foundation on this series. I hope you've enjoyed it as much as we've talked about maternal health, CRISPR, vaccines, all sorts of amazing technologies, all of which you can check out on our YouTube channel. Everything that we do goes live there and stays there forever. So if you want to watch in 3 weeks or 3 years down the road, you can absolutely do so. Now, today I'm particularly excited because we're welcoming in someone who's talking about something that we've very seldom featured here at Exploring by the Seat of Your Pants. Dr. Ed Boyden's joining us from his roles at the MIT as a professor there and the director of the Synthetic Neurobiology Lab. He does amazing work trying to develop new tools and techniques to understand, analyze, and heal the brain. So some really, really cool tech we're going to discuss today. I'm so excited personally to learn all about this. And so without further ado, Dr. Boyden, welcome to the broadcast, and thanks so much for joining us today.
Great. It's great to be here. It's a real honor to get to share with you some of what we were doing. I'm an engineer, so I design and invent things. And in particular, I invent technologies that help us understand and fix the brain. I'm a professor at MIT, and I lead a group of over 50 people, and we're trying to understand how the brain works and how we can help the brain improve. Now, our brains mediate our thoughts and feelings. They make us who we are. But why is it so complicated to understand? Well, the brain is incredibly complicated in terms of what it's made out of and how it looks. So brain cells are very complicated objects. They look like these long, spindly trees. They can be very, very large. They can go throughout our heads in terms of their extent. But their shapes are very complicated, and they're very, very small in terms of their features. So here we just zoomed in to a connection between two brain cells, a synapse. And the synapse is full of molecules. There are about 30,000 genes in the human genome, and those genes encode for huge numbers of molecules that are inside those brain cells that then work like a network, kind of like the internet, that ship information around and compute. But how can you understand something that complicated, right? The size of our head, but made out of things at the scale of molecules? But it's even more complicated. The brain also is very high speed compared to other biological systems. So brain cells create these millisecond timescale, 1,000th of a second timescale pulses of electricity, and they communicate by releasing chemicals that go from one cell to the next at those synapses, those connections I showed you in the previous movie. And those chemical exchanges also occur on the order of 1,000th of a second. And so this is happening all the time. A cubic millimeter of your brain will have on the order of 100,000 brain cells connected by 1 billion synaptic connections. And all those cells and all those connections have those electrical and chemical events happening all the time. So how does this implement things like thinking and feeling, decisions and memories? We can't explain how that occurs yet. But there's another reason to be interested in the brain. Brain diseases affect people. No brain disease can be fully cured. And for many of them, the treatments are quite poor. Alzheimer's disease, ALS, multiple sclerosis, traumatic brain injury. The list goes on and on and on. These diseases don't just change our the time we have to live, or add suffering to our life. They can even change who we are. And so that's one reason why brain diseases, I think, really are so daunting. Treating a brain disease is very difficult. So these are some numbers from over a decade ago. I think they've actually gotten a bit worse. But to take a brain drug out of the lab and into the marketplace where you can prescribe it takes almost 10 years. The failure rate to get government approval is almost 100%, over 90% failure. And the cost will be on the order of $1 billion or more each. And even after all that hard work, even if you do get approval, the effects are partial. Again, no brain disease has been fully cured. So how do we confront these issues of space and time, right? I told you about how spatially complicated the brain is, these complex networks. I told you how temporally complicated the brain is, those high-speed signals that occur. Ideally, we can map and repair the brain across space and time. I'll tell you 2 short stories today, one about space and one about time. The first is about space. How can we see the brain? If you could see the brain in all its complexity, right? Those wires, the connections, the molecules at those connections, maybe you could even simulate a brain in a computer. People like to talk a lot about AI right now. What if you could simulate an actual brain doing actual thoughts or emotions? Unfortunately, this is a hard problem to see a large object like a brain, but to resolve those individual molecules that make it up. I think we might have all seen pictures like this, brain scans. The popular press often features these kinds of images. These are parts of the brain that light up when the brain is active. But those blobs that you see lighting up are gigantic, right? They can contain millions or even billions of brain cells. So it's not enough to map the wiring of the brain. At the other extreme are microscopes. Microscopes magnify images of the brain. But even microscopes struggle to image the brain because light has a finite size or wavelength, and the molecules that make up the brain and the wiring of the brain, unfortunately, is too small for the most part to be seen. So, how can we see the brain? Well, in my group, we often try to do the opposite of what people have been doing. For 300 years, the way you image the brain is you use a lens to magnify an image of it, right? That's microscopy. We started wondering, could you make the brain itself grow bigger? Now, I think we've all, seen baby diapers, right? Baby diapers, when you add water, they swell. And this animation shows you what's happening. So there's a chemical in the baby diaper called a swellable material, sodium polyacrylate, for example. When you add water, the water is drawn in through osmosis, and the polymer in the baby diaper swells. That's how a baby diaper works. We wondered, could you put this baby diaper material inside the brain? Do it perfectly, and you could maybe add water and make the brain grow. Then, inexpensive imaging devices could let you map the brain. We call this expansion microscopy, and it works. We can chemically weave a dense spiderweb like mesh of baby diaper polymer, sodium acrylate, inside brain cells, in between brain cells, around biomolecules, in between biomolecules. Do it just right and add water, and the baby diaper material will swell and make the brain grow with it. So here's a picture of a piece of the mouse brain in panel B. I'll use my mouse pointer to point. I hope you can see it. Panel B shows a piece of the mouse brain. Panel C shows a piece of the brain. About a day and a half later, we form the polymer. We soften the specimen a bit. We add water. And as this polymer swells, the brain physically grows. It's now 100 times bigger in volume. And you can make it even bigger than that. The schematic in the upper left shows how the polymer starts out. It's very dense. It's all jam-packed together inside the brain cells. Of course, this will not work on a living brain, only on a preserved brain. Once you add the water, the polymer swells and you get this extended conformation. So the take-home message here is you can actually start to make a map of the brain. Here we're taking proteins from jellyfish and coral. So jellyfish and coral have glowing proteins. You can express them using gene delivery. This is sometimes called the same kind of stuff that makes gene therapy work. You express the genes that encode for fluorescent proteins, such as those from jellyfish and coral, in the brain. Expand the brain, and now you can start to see the fine wiring of the brain. So here's a piece of the mouse brain involved with memory formation, amongst other things. We express these coral and jellyfish derived fluorescent proteins, glowing proteins, in the brain. We do our expansion microscopy technique, infusing the baby diaper polymer through a chemical process, softening the specimen and adding water to blow it up. And now I hope you can see that the fine wiring of the brain is becoming visible. Imagine now you can make a map of the brain so detailed that you might be able to simulate it in a computer. Could you simulate an actual decision or an actual emotion? Well, that's a job for the future, but it's one of the things that we're very excited about, because if you could simulate parts of the brain in a computer, maybe you could understand really how they work, right? What generates things like intelligence and emotion. And also, maybe it would tell us a little bit about what it means to be human. Why do we feel what we feel and do what we do? Now, the expansion method will not work on a living thing. And so in the second part of the talk, I want to tell you about time. I've told you about space. How do we map the brain across space? Now, let's talk about time. Sorry, the movie's not playing. Okay, time. We have these brain cells firing electrical pulses lasting about 1,000th of a second. Ideally, we could control them, right? If we stimulate them, we could try to figure out what those brain cells do. Suppose you stimulate some brain cells. They might trigger an emotion. They might trigger a movement. They might trigger the recall of a memory. By activating brain cells, you could figure out what they do. Now, how do we do that? Well, if you can try to stimulate the brain using all sorts of older techniques like drugs or electricity, but they all have non-specific effects. So we developed a strategy that we call optogenetics. Opto for light, and genetics because, as you'll see, the molecule that does the trick is genetically encoded. So the basic concept is imagine solar panels that you could install in brain cells. Solar panels convert light to electricity, and electricity is the language of the brain. So if we could put the solar panels in the brain cells, you could shine light on the cell, the solar panel would convert light to electricity, and you could turn it on. Now, how do you get light into the brain? Well, the brain doesn't feel pain, so you can bring electrodes into the brain, even in awake people. This is done in a process called neurosurgery for deep brain stimulation. And so, if you could bring electrodes in the brain, you could bring optical fibers into the brain, connected to a laser, for example. All right, how do we make the brain cells sense light, though? Well, it turns out all over the tree of life, you can find organisms that sense light or that turn solar energy into chemical energy. Here's a single-celled green algae, and it has an eye spot shown in brown. It's swimming around in a body of water. How does it know where to swim? Well, the eye spot is full of light-activated proteins that convert light into electrical signals. When sunlight hits these light-activated proteins, they open a little pore or hole, and charged particles, ions, will flow from one side of the membrane to the other. That's how this single-celled organism knows where the sun is and can swim to the surface of the water so that it can photosynthesize better. Okay, well, it turns out we're very lucky. This protein is encoded by a small gene, a small piece of DNA. And we can take that small piece of DNA and put it into a gene therapy vector, which lets us deliver the gene to another kind of cell. These are the same kinds of gene therapy vectors that are used in all sorts of experimental and increasingly approved treatments for different diseases. For example, the virus AAV is very commonly used. We take the AAV virus containing this gene for the opsin, that's a technical name for the light activated protein, and we deliver the gene into the brain cell. Amazingly, this works. The brain cell can manufacture that light activated protein, and when you shine light on the brain cell, those proteins can convert light into electrical signals. And that's just sheer luck, right? That's serendipity. That didn't have to work. Maybe the protein would have been toxic. Maybe it would not have worked. But we were very lucky, and it worked pretty much on the first try. Okay, this movie has some flashing lights in it, so if you are photosensitive, maybe you should probably look away for this, and I'll tell you when you can look back. Um, but here's an example of how we can use the technique. You can put the gene for the light-activated protein into the brain, and by using gene therapy tricks, you can express the gene just in some cells and not others. The brain has many kinds of cell, right? Some are big, some are small, some activate, some silence, but you can express them just in one kind of cell, like this little star-shaped cell here. Shine light, and these light-activated proteins will convert the light into electrical signals just in the cells that bear our protein. In this case, we're activating a small set of cells that are basket cells or fast spiking cells. They have different names. Now, why is it interesting? Well, my collaborator Li Wei Sai showed that by activating these star-shaped cells at a specific frequency, 40 times a second, in mice that are engineered to get Alzheimer's-like symptoms, these are Alzheimer's model mice, if you activate these star-shaped cells about 40 times a second, the brain gets better. Alzheimer's disease, right? No cure and very few treatments. The treatments also really don't slow down the disease, they just improve the symptoms. But by activating the brain 40 times a second, you can actually clean up the problems of Alzheimer's disease in mice. So now we've been working both in a startup company that we are using to do large-scale trials, as well as in academia, to work on ways to drive these frequencies without using the optogenetics. So Li Wei and her team, with our help, went on to show that you could cause the same brain waves, these 40 hertz brain waves, by having mice watch a flickering light or listen to a clicking sound. No need for optogenetics, right? No need for any gene therapy or optical fibers. They're just watching a movie, but amazingly, the mice got better. So one very powerful use for optogenetics is you can discover brain activity patterns that might help people. Then, of course, you can create those patterns through whatever means is most practical. In this case, by having patients watch movies. So it's an ongoing trial. It's not an approved therapy, but we're very excited about the possibilities. Optogenetics and expansion microscopy are in use by thousands and thousands of research groups around the world. Expansion microscopy is being used to map out how brains and other biological systems work. Optogenetics is being used to study how the activation or silencing of brain activity can be used to help study behavior. You can use it to study brain diseases, and the list goes on and on. Something interesting happened, though, very recently. People started wondering, could you use optogenetics in human patients? A while ago, I was part of a team that looked at blindness. Here's a blind mouse in a water maze. It's a maze with a platform that, if the animal swims to it, it can climb out of the water. And mice like to swim, but they like to sit on a platform even more. This mouse is blind. It's lost its photoreceptor cells, the cells that capture light. And so eventually it can find the platform just by searching, but it's not using its eyes to find it. Now, here's a mouse that got one gene therapy dose of a light activated protein that expresses in the spared cells of the eye. They've lost their photoreceptors, but the rest of the eye is there. And now the mouse can see where to go. It can go to the platform. 2 summers ago, a team in Europe showed that they could take one of our molecules that we reported in 2014 and express this gene using a gene therapy vector in the human eye. These are patients who've lost their photoreceptor cells, but the rest of the eye is still there. And by putting our light sensitive molecule into the spared cells of the eye, they could try to help people see again. So here's a patient, a human patient who had lost the photoreceptors in his eyes and was unable to see. By equipping his eyes with this light activated protein and giving him some goggles to take pictures of the world and project them onto his eye properly, he could start to recognize objects like household objects. He could see the lines of a crosswalk on the street. He could recognize things like doors and so forth. So there's a long way to go, but it's exciting to think that maybe one could try to restore vision in patients with this technique. So that I think I want to acknowledge that this is all a very omnidisciplinary effort. I hope I've showed you that almost every domain of science is having impact on the brain. Polymers like those in baby diapers, studying organisms like those that live in the ocean, gene therapy, and optics. Every part of science is teaming up to try to help us understand and repair the brain. So the work that I described was driven by a long list of people, both in the group at the top and collaborators all over the world in the middle. In such a short talk, I don't think I have time to acknowledge all the people. I'll end on this slide. Our website, synthneuro.org, has more information about our group, and don't hesitate to email me. It's just my name, edboyden@mit.edu. And with that, I'm happy to take questions.
Geez, well, thank you so much, Ed. That was an incredible amount of research covered in 18 minutes. My goodness. A first question we got from a class is, I mean, the optogenetic blindness sort of therapy is astonishing. Is the world doing this? Where are we at in terms of rolling this out, where everyone who has this specific form of blindness can have this as a treatment option, basically?
Yeah, so there's a French company called Gensight that's doing the clinical trials. I'm not involved directly with them. Um, but yeah, I think they're trying to get regulatory approval. They've continued the trials, and yeah, if all goes well, maybe someday it would be a potential therapy.
How exciting is that? We had a great question, which I had too. So when you showcased the fluorescent proteins in the sort of map of the brain, what did the different colors represent? Like, is there a reason why they are shaded the way they are, or how does that actually look in reality? What's the deal?
Those are random. Yeah, we're giving each neuron a sort of a barcode, right? So
Okay.
if you buy a product, it'll have a barcode on it. I don't have an example around here, but I think we've all seen them. What if you give every neuron a barcode so that you can tell them apart? So that's what we're doing. We're trying to give each neuron a color code by expressing combinations of those proteins from coral and jellyfish.
So, I mean, the coral jellyfish is the example. You highlighted the mouse as well. We got a question about animal models for research. You talked about preserved brains. Are there any living animals that you use for brain studies in any way? Because it seems, I mean, a lot of biological research requires the use of animals in some fashion. Is it useful to you? Are there any specific models you can speak to?
Absolutely. Well, the Alzheimer's example that I gave, those were animals that were experiencing the movie, and in Leeway's group, the blind animals that could see and solve the water maze, those were obviously alive. So, yeah, we don't have an understanding of biology, and that's why studying animals or cells and culture or other living systems is so critical.
Yeah. The back to the point of sort of tracing ideas or thoughts in the brain, it's very, very complicated. Do we, I mean, you highlighted this great image of sort of a brain scan when people are thinking about various things, but as you said, that's a very bulk part of the brain. Like it's millions, if not billions of connections in something like that. Do we know if if you have the same thought, cinnamon in your hot chocolate as a sensation, you're trying to bring that up in your brain. Does that thought follow the same pathway every single time in your brain? Do we know that? Are we close to knowing something like that? My festive example, Ed.
Well, everybody's brain could be different, right? I mean, this is sort of almost the realm of philosophy. When you smell cinnamon, how do I know that your sense of smell feels the same as mine, right? Even if we describe it with words, what if you perceive your words differently from me? So some of these things are currently the domain of philosophy. We don't
Yep.
know how to answer these questions yet, but we can try to look at behavior. We can try to look at report. We can try to look at different kinds of function,
Yep.
and you can try to map the brain. So some regions are similar across people. Our visual system is in the back of the brain. Our auditory system might be over here. There are regions involved with speech and with decision making and so forth. But of course, there's also a lot of variability as well. And so that's kind of one of the reasons why we work so hard on technology, right? We want to be able to understand the brain. Um, and we're really limited by technology, as I tried to kick off the lecture with, right? The
Yep.
spatial complexity of the brain, going from molecule to cell to our entire brain, the temporal complexity of the brain, right? Those millisecond timescale events of electricity, how do they add up to form a memory that you might remember 5 or 10 years later? Or, when a disease like Alzheimer's disease occurs, it can take years to occur. How does space and time really add up for the brain? Is something we are just now starting to be able to tackle.
So exciting. I'm sure. I mean, this is the thing that about science that I always like to highlight for our students is that, I mean, it can sound complicated, it can sound challenging. I remember doing grade 11 physics and I was, but when you get at a high level of science, you get to collaborate with people all over the world. You get to work in really cool places. You get to use really cool toys and technologies to do the work that you want to do. And you get to sort of probe the foundational underpinnings of everything in the world around us in the universe. I mean, it's like, it's terribly exciting at its peak, and you get to live that every single day, which is thrilling. On
Yeah,
that
yeah.
question, oh, go ahead. Sorry.
Yeah, it's really a lot about play and discovery. We have a very playful way of thinking about things, right? We're often trying to do the opposite of what people do,
Yep.
and that makes a lot of fun. Everybody's been zooming in with a lens. Let's make it bigger with baby diaper material. People are all trying to use
Like,
electricity to control the brain. Let's use light. I think by thinking differently about things, you can often come up with extremely revolutionary things in a fairly tractable amount of time.
I've got to say, and we'll see what the future holds for you. But when you said the idea of sort of expanding the brain, my first thought was just Nobel Prize. I mean, like that just seems like such a unique take that almost no one would have come up with. I don't know if it was you specifically, a member of your team, or a collaborative sort of discussion, but I mean, that is a really fascinating way of approaching that as a problem. Have you done this with human brains yet? We had a question about this, so I'll follow up with that. Or is it just mouse brains for now?
Well, over 500 studies have come out doing expansion already. It's been used by many groups to study the human brain. It's being applied to plants. It's being applied to bacteria, viruses, you name it. I mean, it can be applied to anything.
Fantastic. What a cool field. We get this question almost every time we've done a Gardner program, and that is, are you using AI for the work that you do? In what way are we using it? How are you using it? Anything that you can share with us? Because people are very keen on artificial intelligence nowadays.
Yeah, I think AI is a tool like any other. We use lasers, we use microscopes, we use bacteria, we use AI. Yeah, I mean, AI can be used to analyze data. You can use AI to generate designs of things like proteins. Yeah, I think it's sort of like air or water at this point. It's everywhere, and it's useful, and we have it as part of our toolbox.
I'm sure the AI companies are thrilled to hear that you link it to air and water and its ubiquity. I like that very much. Um, is there a favorite part of your job? Is there something that you particularly, gets you up in the morning that you love to do most? Is it talking about your work? Is it creating a new experiment? What's the your favorite thing?
Oh, so many parts. Yeah, I mean, it's so much fun to see something for the first time ever. When I saw the first light activated neuron at 1 o'clock in the morning, doing the experiment with my own hands. When we saw the first expanded brain, it almost looked like science fiction.
Yeah.
Yeah, I mean, that's so thrilling to be the first in history to see something is really cool. Um, yeah, but
Yeah.
everything is has parts that are fun. It's great to see our students go on and become leaders of new things. Many of my group's alumni have now gone on to lead organizations that are trying to revolutionize climate change or to revolutionize our ability to deal with aging, or
Yeah.
or we have a student who started a company that's trying to revolutionize 3D printing of nanotechnology. List goes on and on.
For your grad, it certainly seems to. That's incredible. I love that you focused on something that I don't think any of our other Gardner laureates have talked about, but it's something that we hear from paleontologists all the time, deep sea biologists. Like, you're the first in the world to see something. There are so many disciplines of science, whether you're doing deep space mapping, going to the ocean with a robot for the first time, cracking open a stone and finding something, or doing an experiment to see something with an idea that you came up with on an approach to a new problem. That's a very that should be the slogan for science going for people going to university. You can see things that no one has ever seen before in human history. Very cool. You touched upon this a little bit, but it's a question I figured we might get. Are we close to creating a robotic brain? Can we create an intelligence that is like ours or even comparable at this point, from the neuroscience side of this equation rather than the roboticists?
Well, we don't have the data of any complete brain yet, right? The molecules, the wiring, the activity. We're trying to put together a large team to do that for the first time, to get comprehensive data sets from our brain, all the activity, all the connectivity, and all the key molecules. And then our goal is to put that data into a computer model and to make the first biologically realistic computer simulation of a brain. But it hasn't been done yet, and I think the reason is until tools like ours have come along. I mean, you just don't have the data, right?
Yeah. Is there a timeline? I know people don't like to be held to these things. Do you have an idea of if we're 10 years off, 50 years off from something like this? I mean, the field seems to be expanding rapidly. There's a lot of amazing labs involved in this work. There's a lot more investment than there seems to have ever been before. I'm curious if you think that we're close or if we have a lot more steps on the way before we can even think about something like that.
Well, this is what I like to call real science. We don't know for sure what the answer is. If we did, I wouldn't think of it as like, you know, true science if you completely know the answer to the question. Um, yeah, but we're part of the team that's putting together some various plans. And yeah, my hope is that it could be done in some reasonably small number of years. So we have the data, maybe optimistically 3 years, pessimistically maybe 10 years or more, but hopefully not out of that range. I mean, who knows for sure though, right? This is what I like to call real science, and there are unknowns, right?
I appreciate the nuance and the fairly soon pessimistic timeline if the nuance is on the better side of things. It's exciting. We've just got two more questions for you. Thank you again so much for your time. I know you do give a lot of talks, so we really appreciate you joining us today. But we get this question a lot as well. Is there the biggest challenge to your work? Is there something that is really tricky, that is a consistent problem, whether it's an approach to problems or something right now that's bothering you? However you want to interpret the biggest challenge question, up to you.
Um, well, the biggest challenge I've faced throughout is, hmm, I guess it's persuading people that the plan is good, because the plan often involves luck, right? I mean, the optogenetic molecules came from nature, right? The diaper expansion method is so different from how anybody had done microscopy before. So, yeah, when I was looking for a job, I had already, uh, you know, co-published this optogenetics paper in 2005. That's what we got the Gardner Award for. Um, but I don't know. People might have thought it was too good to be true. People thought, nobody's ever started a group that just does brain technology and nothing else before, and it was hard to get job offers. So yeah, that's how I ended up in a department within the MIT School of Architecture of all places. And yeah, most neuroscience departments and engineering departments rejected my job application, and so that was tough. And then when we eventually, of course, people got persuaded that the technique works, and now thousands of people use it. But then when we did expansion, also I think people didn't believe it. I wrote like 9 government grants trying to raise money for it, and yeah, we almost went bankrupt there as a group, which was quite depressing. But thankfully, because of a long chain of random events, word got out that we had this great idea, and it was hard to get money for it. And a foundation called Open Philanthropy ended up giving us a gift to support the work. So, time and time again, I think the challenge is persuading people that these really crazy sounding ideas are good. And often the way that we achieve it is through luck. So I started really wondering about this idea of, is luck a learnable and teachable skill, right? Optogenetics coming from nature, or expansion microscopy being a collision of disciplines, or funding coming from unexpected sources. Can we teach people how to be lucky?
Yeah. What an interesting that there's a whole chapter of your future book right there with that answer. That was spectacular. And it's funny how often luck plays a role. We bring on people from all manner of disciplines. And again, pretty much everyone will tell you, they had a lucky break. They took the chance to talk to someone. Oh, they found a mentor that really changed their life. Oh, something came through at the last minute. This is not an uncommon story. And I think it's really kudos to you for emphasizing that in your own career trajectory. And that's very exciting. We were talking about vaccines a few weeks back, and Catalin Carrico just won the Nobel Prize for the RNA work, talked about the same thing. I mean, no one took her seriously. It was hard to get funding. It was hard to get a job. Nothing was really secure. And now she's one of the most lauded scientists in the world. And this seems to be the case for a lot of cutting edge technologies, that people are distrustful and sort of leery about things that are wildly different than anything people have tried before. But those moonshots can often lead to some really spectacular development. So thank you for that way of phrasing it, Ed. All right, one final question before we wrap up, and I always like to wrap up with this. For our students that are tuning in, we've got high school students, we've got late middle school students that want to become you. Is there something you'd recommend? Are there courses they should take, resources that they should look into, anything that really impacted you as you were growing up into this field that you'd recommend to people that want to follow in your footsteps?
I think it's really important to know the fundamental sciences very solidly. I think of the brain as a computational system that runs on chemistry according to the laws of physics. I sort of a weird path. I ended up as an undergrad for 6 years. So I spent 2 years as a chemistry major and switched schools, and spent 4 years studying physics and also electrical engineering and computer science. And so learning these fundamental knowledges is so powerful, because 20 years later, 30 years later, that knowledge is still true, and you can apply it to problems like biology. So I really think learning the fundamentals very solidly is very powerful. And at
Interesting.
some point, you'll have learned all the skills that you think you need, and then you can pivot towards impact, right? And there, I think the key is to really think from a ground truth perspective, right? What are the fundamental things and how do they work? So if you think about chemistry, right? After the periodic table was discovered, now you can make all sorts of different molecules, right? Because you know the building blocks, atoms, and how they work together, reactions, right? Physics, right? So before quantum mechanics, it was very hard to do much in physics. But after quantum mechanics, now you got the microchip and the laser and the computer and the internet and the cell phone, right? So, and again, that's because quantum mechanics is the building blocks and how they interact. So, yeah, so I think going after a fundamental description of what is mysterious is one of the most powerful strategies in scientific history.
Yeah. What an interesting answer. I've never actually had someone answer it that way. And I love it, because one of the things that we like to emphasize in a lot of our broadcasts are that really innovative governments throughout history, companies throughout history often provide the opportunity for people of wildly different disciplines to interact with one another. It's a different way of thinking. You have different people with those fundamental bits of knowledge that can partner and create something that's unique. And your educational background sort of mirrors that, but it's all within you. So if people have that opportunity, I guess it's sort of a call to action to be at least a generalist in terms of getting that fundamental information. And I really think that's a cool message for our kids. So Ed, this has been spectacular, man. Thank you so much for joining us today, sharing all this. I'll be hawking this to the Gardner team and many more people in the days to come, because this is a really fun broadcast. But thank you for joining. Is there any last message you want to share with our kids before I say farewell and end our program there?
Um, Well, advice is always of a given time and from a given perspective. And one bit of advice I gave myself many years ago is anytime somebody gives you advice, at least visualize and imagine what would happen if you did the opposite of it. Because, science is about doing things that are new. And that's maybe I'll end on there.
Uh, Ed, you've given me some of the most unique answers we've ever had in the history of Exploring by the Seat of Your Pants, and I've hosted 1,500 of these, so that's hard to pull off. So thank you for that. It speaks to your work as a scientist more broadly, and I really appreciate it in the science communication context. Classes, thank you so much for tuning in. Again, this will be on YouTube forever if you want to watch anytime. Check it out on our channel. Check out Gardner Foundation's website to see all the amazing people that they laud and feature there. And Ed, without further ado, have a wonderful rest of your day.